Welcome to the Cell Communication Series. This video focuses on receptors. At the end of this lecture, students should be able to understand how signals interact with receptors, differentiate between types of cell receptor, and describe intracellular receptors. There are many different forms of cell receptors. Some respond to signals based on a mechanical motion change. These are called mechanoreceptors. There are also receptors that respond to signals resulting from pressure change. These are called baroreceptors. Receptors responding to temperature sensitivity are thermoreceptors. And light signals are received by photoreceptors. The form we will focus on are chemoreceptors, which adjust to changes in chemicals. Often, the signals trying to transcend the plasma membrane are small, but hydrophilic. They may be small or charged, or too large to pass through the membrane. These extracellular signals bind to receptors to get their message across. Hundreds of receptors are embedded in the membrane to connect with these various types of signals. There are three main receptors we will talk about. The first are enzyme-linked receptors. The second are G-protein-coupled receptors. And the third are ligand-gated ion channels. The first type of receptor is known as enzyme-linked receptors. These receptors transverse the membrane and with an extracellular domain on the outside and an intracellular domain on the inside. Typically, hormones like insulin can bind to these receptors. They are considered enzyme-linked because in the internal environment, the receptor leads to a catalytic function. The signal binds on the extracellular receptor, leading to a conformational change in the intracellular side, and that leads to activation of the catalytic domain. Remember that protein kinases cause phosphorylation of proteins. The signaling molecule binds to the extracellular domain of the inactive receptor. This causes the conformational change in the receptor, leading to the intracellular domain to pull the highly energized third phosphate from ATP and adding it to the protein, giving the protein a new function. In the case of insulin, the protein being phosphorylated is tyrosine. This creates a signal transduction cascade that leads to an increase in the uptake of glucose from metabolism. Signal transduction is a way that the primary messenger or signal activates proteins like tyrosine, which then work with other proteins to relay the signal to transcription factors that then create a cellular response by turning on or off specific genes. The next receptor is a G-protein-coupled receptor, sometimes referred to as GPCR. Eukaryotes, and animals especially, use GPCR. Instead of using ATP, these receptors activate a mechanism using GTP, which uses the nitrogenous base guanine in lieu of adenine. So instead of adenosine triphosphate, it's guanosine triphosphate. As the G protein couples to the receptor, it binds the signal. The receptor consists of 7 subunits woven throughout the membrane. This receptor, once bound to the signal, will change conformation, and it activates the G protein complex. This causes a release of GDP. It now binds to GTP instead from the cytoplasm and changes its conformation again. This releases the alpha subunit of the G protein. It can trigger the signal transduction pathway. The alpha subunit gives off the third phosphate, allowing it to reassociate with the beta subunit in the G protein complex. The final receptor is a ligand-gated ion channel. When we learned about membranes, we found that ion channels allow the passage of charged particles across the membrane. In this case, the signal molecule is referred to as the ligand. The receptor is closed to the extracellular fluid until the ligand binds. Once 2 ligands bind to the receptor, the channel opens, allowing ions to cross the membrane. These are especially important with synaptic signals between nerve cells. Some receptors are intracellular or found within the cell. These receptors can only respond to ligands that pass through the membrane. The endocrine system capitalizes on these mechanisms since steroid hormones freely cross the membrane. Estrogen is a steroid. This hormone travels all the way to the nucleus to meet its receptor. Once bound to the receptor, it changes the conformation and binds with another activated receptor. The estrogen complex dimer binds to DNA, and this leads to a transcription of genes in order to express proteins, or to make genes that change the cellular structure or function. This also changes how the cell responds. Students should be able to describe how signals interact with receptors. They should be able to distinguish between three main types of cell receptors. They should be able to discuss intracellular signaling and receptors.